So I'm going to go into my explain everything here. All right, so what I have done here is I've drawn some pieces to our DNA protein synthesis model. And I just realized that something I made a change to didn't stick. So maybe I'll get you guys to help me figure out what these amino acids are in just a moment. But before we get to what these things are and what happens outside in the cytoplasm, I want to point out that biology has something that we call a central dogma. And what a dogma is it's a belief system, it's a principle. And so it's basically what the basis of biology is. It's the foundation of biology. And what it says is that DNA gets converted into RNA, and then RNA gets converted into a protein. Okay, so you can't go from DNA to protein without going through RNA. And these arrows represent the process that occurs when DNA gets converted into RNA. So this arrow right here is representing transcription, the process of turning DNA into RNA. And then this arrow here represents translation, when we take the RNA code and we turn it into a protein. So yes, DNA does code for proteins that your body needs, but it can't do it without RNA. And so this whole system that, this whole process that Mr. Austin's been going over is how our bodies do this, how the code of DNA gets transferred into RNA and then turned into proteins in our bodies. So I'm going to start at the very beginning with DNA. And if I'm going to talk about DNA, I'm going to have to start in the nucleus. So I'm going to draw for you a nucleus. And remember that the nucleus is where DNA is kept. And DNA is double-stranded. It's in the shape of a double helix. And the DNA is really big. It's long, it's cumbersome with its double strands, and it cannot leave the nucleus. Okay, it stays in the nucleus where it's kept safe. You don't want to break or lose parts of your DNA. That's really bad. So the nucleus is like a protective place for your DNA to stay. And so that's why that RNA piece is so important. So a process happens inside of the nucleus where the DNA gets turned into mRNA, which is a single-stranded, and it's not as long. It only carries the code for one protein, so it's short. It's much shorter than the DNA. And this process is called transcription. And you learned a little bit more about transcription, but the basics of it is that one strand of the DNA gets coded for, the template strand, right? And that's what gets turned into mRNA. And there's an enzyme called RNA polymerase that takes part of this. And there are some pieces of transcription that you needed to understand, but that's when the DNA is converted into mRNA. Now, this mRNA after transcription is not quite ready to leave the nucleus, because it has to have a few things happen to it first. Okay, it needs to be what we call edited. And so Mr. Austin spent some time teaching you about how there needs to be a cap that's put on the 5' end, right? This is called the methylated G cap. I'm going to abbreviate methylated. The methylated guanine cap. And on the 3' end is added what we call a poly A tail. And poly means many. And this is no joke. There's like 200 to 500 A's that get put on the 3' end. And the reason for that poly A tail is because when the mRNA leaves the nucleus, that tail, some A's might fall off. And they fall off while the mRNA goes out into the cytoplasm. But because there's so many A's, it's okay if it gets lost. Parts of it get lost. What's important is that the code for that protein that's in between the G cap and the poly A tail, that's kept safe. Okay, so this G cap keeps the front of it safe, and the poly A tail keeps the back end safe, because this guy has to go for a little journey out into the cytoplasm. All right, and then I gotta make sure that we understand too that there are letters that are not supposed to be there. So transcription copies all of the DNA letters, but there's some letters in the DNA that are introns, they're in the way. And so they have to be cut out. And you learn how to do this using the codon wheel, how to cut out those introns. So I'm just gonna, those X's are representing my introns that I had to cut out of the mRNA before it can leave the nucleus. All right, so now everything's ready. The mRNA has the cap, it has a tail, the introns are cut out. Now it can leave the nucleus, and it leaves the nucleus through these holes. I'm going to try to make them look 3D for you here. There's holes in the nucleus called nuclear pores, kind of like you have pores on your face. If you look really closely, like little holes in your skin. There's pores in the nucleus, and they're big enough to let the mRNA out, but they are too small for the DNA to come out. And so this mRNA then takes a journey, and it goes, 5' end goes out first, and it leaves through this nuclear pore out into the cytoplasm. So the mRNA takes a little journey out into the cytoplasm, where now the next step of this process is going to happen. Okay, so up until now, all we've talked about so far is DNA going to RNA. So any questions on this process right here? Is everyone feeling good about the first steps of protein synthesis? All right, Caden's feeling good about this. I hope everyone else is feeling good about it. All right, so now that we're out in the cytoplasm, this is where things get a little bit trickier, because we have a lot more details and different moving pieces happening. So what I have here is my mature mRNA strand right here. It has its codons, this 3-letter code that codes for amino acids. It's got its tail, it's got its cap on. So this is going to be read and turned into a protein. The other pieces I have here are my ribosome. So this green thing that kind of looks like I'm starting to draw a Daffy Duck is representing my ribosome. And it has 2 parts to it. It has a small subunit and a large subunit. And what happens is the mRNA gets sandwiched in between the large and small subunit here. And then it has 2 sites that Mr. Austin talked about, the A site over here on the right and the P site. Okay, and these are places where the tRNA are going to fit into the ribosome. All right, out here I have my tRNAs floating around, and they float around in the cytoplasm carrying amino acids and getting ready to deliver them to the ribosome. So I want to point out that our tRNAs have two distinct parts to them. So down here at the bottom, they have what we call an anticodon. And the anticodon is really, really important because it is going to match up. It's a complementary pairing to a codon. Okay, and so this anticodon tells the tRNA where to bring its amino acid. So this circle up here is representing my amino acid. And when I went through, I originally went through and labeled all these amino acids, but apparently I didn't save this correctly. When I went to label the amino acids on the top of this tRNA, I can't use my anticodon. Okay, I have to think about what the codon was for this that matches with this anticodon so that I can use my codon wheel. So in the chat, will somebody tell me what the codon would be for this tRNA anticodon? If it's UUA, what would my codon be? Oh, Foster, be careful. We're talking about RNA now, right? So we can't have any T's in there. We left the T's in the nucleus, right? Yeah, AAU. So I'm going to look at my codon wheel. AAU is asparagine. Asparagine. And I'm going to do another one here. What would this codon be? If it's GAU anticodon. Somebody just asked me if you're allowed to use the codon wheel on the test. Absolutely. We would never make you memorize the codon wheel. Oh, Aiden's telling me it's leucine. So that would be CUA. CUA. Yep, you're right. You jump to the next step, Aiden, which is awesome. So Aiden had to first convert this anticodon to CUA and then use his codon wheel, which you did perfectly. Nice job, Aiden. All right, let's label this one here. If it's UAG anticodon, what would be my amino acid? This does require you to have your codon wheel handy. Anyone? So first we have to think about what the codon would be, and it would be AUC. And so then going to our codon wheel, AUC, and I have a direct message from somebody saying it's isoleucine, which is correct. All right, so I think I'm going to just quickly label these other two. So GAA would be glutamic acid. And GGC would be CCG, which would be proline. All right, so now we're ready to start this process of building this amino acid or this polypeptide chain. Let me quickly just erase this because otherwise it will move with me and I don't want it to. I don't know if anyone's used Explain Everything, but it's really cool, but there are some limits to its abilities. All right, so first of all, I'm gonna have to move my ribosome into place. And remember that we talked about how AUG is the start codon. And so the initiation of protein synthesis occurs when the AUG is moved into the A site of the ribosome. So the ribosome attaches at the 5' end, and the AUG is in the A site of that ribosome. Now we have our methionine, our tRNA here, that's going to come in, and the complementary codon is going to match up with that anticodon, and the ribosome is then going to move one codon over to the next set of 3 letters, CUA. So what is my tRNA that's going to come in? What is the anticodon to CUA? And Amelia is telling me it's GAU, which is up here. So in comes leucine. Now, once there's 2 tRNA molecules together like this, now a peptide bond is going to form in between these 2 amino acids here. So there'll be a peptide bond between methionine and leucine that gets formed. And then the ribosome is going to move over one codon at a time. It always moves one codon at a time. And then the next tRNA is able to come in. And that next one is going to be UAG, right? Isoleucine. So here's where my Explain Everything starts to kind of lose its ability to show this to you. So these red bonds right here that I'm drawing in, these are peptide bonds, right? That are forming in between the amino acids. And technically this tRNA over here that was connected to methionine, this would now leave. The tRNA would leave empty, and it would leave methionine behind. And so the best way for me to show this to you is just to erase the tRNA, even though it doesn't disappear like that. It actually leaves and goes and finds another methionine. And then the ribosome is going to move down to the next codon. And see, this is where my this is where Explain Everything is amazing, but it also doesn't let me. I wish it would leave things that I drew behind, but it actually moves them with me. All right, so I hope that you guys are starting to see kind of the process that's happening here, that peptide bonds are being made, and the next tRNA would come in. If it's AAU, it would be this UUA, and another peptide bond would be made. And this is just going to keep on occurring until the ribosome makes it to what we call a stop codon. And so we talked yesterday about UGA being a stop codon here. I just want to make sure you guys understand there are 2 other stop codons. UGA is not the only stop codon. Okay, if you look on your codon wheel, there's 3 different locations that you can find a stop. Okay, and what this means is when the ribosome hits this code, it doesn't bring another tRNA in, it brings in what we call a release factor. And it fills in that A site on the ribosome. And I like how Mr. Austin explained it's like an anchor. It doesn't allow anything else to be added, and it makes the ribosome come apart, and it releases the protein. So that protein then, once the release factor has disassociated the ribosome leaves, and what we're left with is, and I'm just going to redraw this because my explain everything has gotten a little crazy here. What we're left with is a chain of amino acids that we call a polypeptide. And you've probably heard us calling it a protein as well. It's not technically a protein until it gets folded and processed inside of the cell. And then once it's made, that protein is going to go on to do whatever it is that your body needs it to do. So for instance, if it's an enzyme that helps you digest your food, it will go into your digestive tract to do its job. If it's hemoglobin, which is a protein that helps carry oxygen in your red blood cells, it will go to your red blood cells to start picking up oxygen. If it's, if you've been working out and your bicep needs more protein, needs more muscle because you're building your muscle in your arm, that protein will go and become part of your bicep. So your cell knows what it needs to make. The DNA tells the cell when to make that protein, and then that protein will leave the cell and go do its job. All right, I'm seeing the chat light up a little, so I'm going to check to see. Somebody asked me, how long does this whole process take in our bodies? I'm not exactly sure about the exact timeline, but it's pretty amazing how quickly proteins get made from an mRNA strand. It doesn't take very long at all. And then I think transcription happens fairly quickly as well. So, and remember, you have millions of ribosomes in your cytoplasm. So this is happening in many, many different locations within each one of your cells. It's not just happening at one ribosome with one mRNA. Your cells are pumping out proteins by the millions. So, it's pretty amazing, really. So I hope that made sense. I'm sorry that my explain everything ribosome got kind of crazy here. But I hope that it helped you understand this whole process from the DNA in the nucleus and then out into the cytoplasm where this second process of translation happens. All right, I am going to stop sharing my screen. And I'm gonna see if, does anyone have any questions about any of those things I just went over? I hope it